So my name is Scott Allen. I'm a lecturer at the Sheffield Institute for Translational Neuroscience. So we are a designated research institute focusing on neurological diseases, such as Parkinson's, Alzheimer's and motor neuron disease. One of our major remits is really to try to translate the findings we find on the bench to real clinical benefits for people with these devastating disorders. So one of my major avenues of research is looking to see how hypoxia, low oxygen conditions, affects the metabolism of people with Alzheimer's and motor neuron disease. So there's a lot of evidence to suggest that people with motor neuron disease and Parkinson's have reduced ability to make energy, and that hypoxia, low oxygen conditions, can play a really important role in that. And there's evidence in the literature showing that brain cells called astrocytes, which are really important metabolic support cells for neurons, are affected in hypoxia and Alzheimer's disease, and also motor neuron disease as well. Motor neurons disease patients tend to suffer from diaphragm weakness, which can cause intermittent bouts of hypoxia, and there is evidence in the literature showing that firefighters, for example, are twice as likely to develop motor neuron disease compared to the normal population. So historically, we've really been limited in our ability to measure metabolism under hypoxic conditions due to the lack of available technology. And so we recently bought a Whitley H35 chamber, and that allows us to take our patient models of disease, assess how hypoxia affects metabolism. So we can take skin cells from patients with motor neuron disease, for example, and reprogram them into brain cells called astrocytes. And then we can expose the astrocytes to levels of hypoxia and then assess how the metabolic profile of these cells is altered using a COS Bioanalyzer. So we've been using a COS Bioanalyzer for 6 or 7 years now, and what this machine allows us to do is it allows us to measure the two major energy generating pathways of the cell, the mitochondria, which is the battery of the cell, or sugar oxidation called glycolysis. And what the machine does, it creates a microenvironment on top of those cells, and you can measure energy production in real time. And so combining this technology with the I2 chamber allows us to do the same measurements under hypoxia. And there's lots of literature evidence suggesting that hypoxia can affect certain pathways in the cell. It can upregulate other pathways and downregulate corresponding pathways. But what we've never been able to measure is how diseases such as MND or Alzheimer's disease affects the levels of these pathways. Now with our purchase of the Whitley I2 chamber, we can. So what we've been able to do in the past is do gene expression experiments where we can expose these cells to hypoxia and then measure the effects of genes. But genes only give you the potential of a system. They don't tell you a great deal about function. And so we have a recent piece of work in Alzheimer's where we'll be able to test gene changes under functional levels in our I2 chamber. And what we've been able to show is that the gene changes match the functional outcomes of these cells, and also being able to measure really subtle changes in metabolism that would never have been able to predict using gene transcription analysis. So what we tend to find as science is that scientific advancement goes hand in hand with technological advancement, and now we have this ability to measure and assess how hypoxia affects the metabolic profile of neurological diseases. We should be able to target pathways for drug screening or target pathways for gene editing where we can try and correct the dysfunction and hopefully improve the quality of life of people with Alzheimer's and motor neuron disease, and also extend their life as well.